In the previous lecture, we talked about different ways of communication between neurons, and what we're going to discuss in this short lecture snip would be how does action potential or electrical events in the presynaptic terminal lead to the release of neurotransmitters. We are going to learn about different voltage-gated channels, and we're going to talk a little bit about the original experiments that have really shown us that calcium regulates neurotransmitter disease release, and that will also lead to the theory of quantum nature of the release from the synaptic vesicles. So the first question that they asked was, what are the signals at the presynaptic terminals that lead to the release of the neurotransmitter? So this is the experimental setup that Bernardo Katz and Riccardo Miledi used in 1967. It is the giant synapse of the squid, which is really large enough that you can insert electrodes in both pre- and postsynaptic elements. So here we have the voltage recording and the current passing electrode in the presynaptic structure, and then we have in the postsynaptic structure we have voltage recording. So what is happening is that when the presynaptic neuron is stimulated, it will fire an action potential, and then after a brief delay, EPSP, or excitatory postsynaptic potential, is recorded in the postsynaptic neuron. So what they have been really wanted to ask is, we know that action potential is produced by the influx of sodium into the neurons and efflux of potassium through the voltage-gated channel. So they first asked whether either of those changes in these ions, either in potassium or sodium, are the signaling that triggers the release of neurotransmitters. So in the first experiment that you have here, what they were passing the current through the electrode, so they were depolarizing the presynaptic terminal, and then they were recording this depolarization, and then recording the postsynaptic. And in this case, the amount of neurotransmitter that's really released is measured actually by the size of this postsynaptic depolarization. So when you measure the postsynaptic membrane changes, membrane potential change, that's our measure of the neurotransmitter release. So in the first experiment, they used tetrodotoxin, which is a blocker of sodium channels, and they again would, in the bath solution, using this setup, they would add tetrodotoxin, they would depolarize the presynaptic membrane, and it still led to the changes in postsynaptic membrane depolarization, which suggests that sodium channels or sodium influx into the cell is not the signaling that leads to the release of neurotransmitter. It is important in normal cases to depolarize the presynaptic membrane because they knew that this depolarization is really required. As we said, there is also the efflux of potassium, so they want to see whether this may be the signaling. So they just added, in addition to tetrodotoxin, and they added tetraethyl ammonium, which is a blocker of potassium channels. Again, they would depolarize presynaptic membrane, and then this would lead to the change of the postsynaptic membrane depolarization. What is really important in both of these experiments is to note that the amount of current would produce larger stepwise depolarization, which would really be reflected in stepwise depolarization of postsynaptic membrane. So there is a relation between the depolarization that we see in the presynaptic membrane and postsynaptic response, which was shown here. You pretty much need to reach a threshold of presynaptic depolarization of 40 millivolts in order to see response in the postsynaptic membrane. So they knew that there is voltage, the depolarization of presynaptic terminal is really important. They just did not know how it be. And they also proved by these experiments it's not either through the influx of sodium or efflux of potassium that plays a role. Channels that they turned to are voltage-gated calcium channels. Now, they knew that voltage-gated calcium channels exist, but they were sparsely present in most places except at the presynaptic terminals. So here is a freeze fracture showing at the presynaptic membrane. This is neuromuscular junctions in the frogs, which shows a row of calcium channels. So there are quite a lot of calcium channels that are present there. I'm just going to briefly give you the overview of voltage-gated calcium channels at the active zones that you will be probably learning much more in your other classes, but will be important for you to know for the future understanding of how plastic these chemical synaptic transmission is really important. So there are 5 different calcium channels. We have L types, PQ, and NR types. These are all high voltage activated channels, so they need voltage that needs to be positive to minus 40 to minus 20 millivolts in order to be activated. So that's where the HVA comes designation. And then there are also T-type channels, which are low voltage. What is important, different channels are present at mostly at the neuronal terminal. We have PQ and N types, and each of these different channels have different toxins with which you can then test their role in the synaptic transmission. So we know that PQ and N type that I told you are going to be inhibited by different toxins. For example, P and Q are blocked by agatoxin, which is spider venom, and N types are blocked by conotoxin, which is a cone snail venom, so you can use them. What also I want to show you, so in addition to using different toxins to test their definite role, what is important also to know their structure. They have the voltage, so this is their voltage sensors. So they have 4 different transmembrane domains, 4 different subunits or subparts of one molecule. So this is the alpha subunit, which has again 4 different repeats of 6 transmembrane domains, and that forms the majority of the channel. However, its activity is really modulated both by the other subunits such as beta, alpha 2, and gamma subunits that also come to form a channel, but also by different kinases in the cell, such as protein kinase C and kinase 2, which we will be talking in a few lectures as well, G protein signaling, which can also lead to the changes in these activity of voltage-gated calcium channels, and also there are subunits such as B that interacts with the protein that are enriched at the active zones to ensure their enrichment at the active zones. Okay, so now this is what we know right now, but then we will go back in time to see how the role of voltage-gated channels has been proven playing any role. Well, what was known from the 50s is if you reduce the extracellular calcium, this is going to mostly then result in the failure. So you will stimulate the neurons. So this is the experiment, the original experiment, and then this is the summary of that experiment. So when you soak the muscles in reduced calcium concentration, so 1/4 of your normal calcium concentration, and then you stimulate, most of the cases actually will get no response. So majority will be failures, and then occasionally we'll have response, and that would be really graded. So this is showing you amplitude of these potentials, and so they will be increasing these stepwise fashion. But pretty much reducing the calcium on the outside prevented or diminished the size of membrane changes potential in the muscles. So when they measured, so when you add both tetrodotoxin and tetraethylammonium, so you're blocking both sodium and potassium channels, then you can measure just the calcium current at presynaptic. So again, when you induce the current, when you depolarize the membranes, you can measure the presynaptic calcium currents, which again were increasing in a graded fashion as you would depolarize more the presynaptic membrane. And then this really correlated very well with the postsynaptic potential. So more of calcium current was entering, you would have increased postsynaptic potential. So this experiment in 1977 said that neurotransmitter release, again here measured as a postsynaptic potential change, correlates with the presynaptic calcium current. This is just the correlation. The calcium, this precise timing of presynaptic calcium was also shown at the central synapses. In central system, it's a little bit harder to measure because presynaptic and postsynaptic terminals are small, and you have many presynaptic terminals contacting neurons. But there is a specific synapse called in the calyx of health. So this is all presynaptic neuron terminal, and then this is your postsynaptic cells. It's a large synapse in the mammalian brainstem, which is playing a role in auditory pathway, and that's where they were able really precisely time the events that happen during chemical transmission. So we have presynaptic potential, so you will activate or depolarize presynaptic terminal. This will lead, then you can measure the calcium current in the postsynaptic cell, and you can see that there is this delay between increasing presynaptic potential to the calcium current. And then pretty much as this calcium, you will have a postsynaptic current, pretty much instantaneous here. And this told us that the major delay between presynaptic potential and postsynaptic action potential, which we call, if you remember, a synaptic delay, is mostly due to this presynaptic calcium current or opening of calcium voltage channels. And that pretty much instantaneous as you have calcium, you will release the neurotransmitter to lead the postsynaptic current. Now, finally, to really prove that calcium entry that correlates so well with the release of neurotransmitter is what is required for the release of transmitters, you need to block it. So what was done in this experiment, and again we are back to the squid vagina synapse, in the similar setup at the beginning, it was exactly the same. So you have presynaptic membrane potential leading to the calcium current and postsynaptic membrane potential, and then you can add different calcium channels blocker. As I have told you, different channels, calcium channels, are going to be present at the presynaptic neurons, and you can add the specific channels. So by blocking the calcium channel, you will block the calcium current, and that is sufficient to lead to completely block the change in postsynaptic membrane potential, which really suggested that the calcium entry through the calcium voltage gated channels in the presynaptic neurons is required for the release of neurotransmitters. What these experiments have shown so far is that opening the calcium channels and then the calcium current that gets calcium that gets inside somehow triggers a release. But how does this influx of calcium trigger release? Now, Katz and his colleagues provided the key insight into this question by these experiments that we took before, and again, in which they decreased the calcium, extracellular calcium, and they showed this stepwise changes in the EPP, which then led them to suggest that transmitters release in these discrete amounts that they called quanta. So each of these successive peaks would be more quanta released. And then this coincided really with electron microscopy images, which have shown the synaptic vesicles and the presynaptic terminal, and especially presynaptic vesicles in the process of fusion or release of neurotransmitter, that then suggested that these might be the physical manifestation of the quanta. What is important to really realize is that the calcium that enters inside of these presynaptic terminals is really concentrated at the active zones. We already have talked about presynaptic calcium channels being focused at the active zones. And this is showing actual neuromuscular junction in which they're perfectly opposed for the postsynaptic receptors. And they are really close to these synaptic vesicles again. We can also visualize the calcium entering through these calcium channels. You can use different calcium indicators. For example, in this experiment here, they use calcium sensitive diuretic 2, but they're also genetically encoded indicators. So you can see that this is a presynaptic terminal, and then they look at the presynaptic calcium, and warmer the color again, there is increased concentration of calcium. So you can see that the calcium is really locally increased when there is an opening of these calcium channels. So it leads to a really local increase in the calcium concentration that is perfectly opposed from the postsynaptic acetylcholine receptors. In the central synapses, we know much less about the calcium signaling, but there are different models that talk about calcium, how it will be the amplitude and spatiotemporal distribution of this signaling. We know that there are interplay between calcium influx and then also calcium removal mechanism through diffusion and buffering. So we know that extracellular calcium is around 2 millimolar, intracellular calcium is really low, at rest is around 0.1 micromolar, so we have a huge driving force to get calcium inside of the cells. When the calcium channels open, the calcium can spike really quickly, up to 100 micromolar, so it's a huge change in its calcium within less of 10 nanometers from the calcium channel mouth, very quickly, within 1 millisecond. So this is another model in which we have shown different vesicles, and then here are calcium channels that open, and you can see very quickly, within 1.15 milliseconds, a huge increase in the calcium concentration around the mouth of these channels. And then how this spreads the further away from the channel you are. So already here we have 8.7 microns, while here we have 20 micromolar concentration of calcium. Why this happens? Because calcium that moves away from the channel will get buffered really rapidly by the calcium binding proteins or diffusion. So it's stressing really importance of the short distance between calcium channel and the vesicles that can then fuse and release neurotransmitters. What we will be talking in the next lecture is how calcium ions lead to this release of neurotransmitter by binding to the vesicles. Just to summarize, what we have talked so far is that calcium enters the presynaptic membrane, and then somehow this leads to release or fusion of the presynaptic vesicles with the membrane. In the next lecture, we are going to get way more acquainted with this mechanism, with the molecular machinery of the fusion presented here, but also the other proteins such as calcium sensor in the vesicles and the proteins at the presynaptic or the active zone that are involved in this role. Now, what I also want you to think about at home after you listen to this, remember I told you that how close the calcium channels are to the vesicle is really important. So this is your question, which we'll talk about it. For example, the way you can assay for calcium diffusion distances, you can use exogenous buffers such as BAPTA and EGTA, which will chelate calcium, to provide information about the spatial organization of calcium channel and releasable vesicles. So, for example, in the squid giant synapse, EGTA, which is again slow, has no effect. But if you use 10 millimolar EGTA in the presynaptic cytosol of the calyx in the mammalian CNS, this will reduce the average release probability of vesicles to about half. So again, in the squid, it has absolutely no effect, while in the mammalian CNS, it will reduce it about half. So what this tells you about the average diffusion distance between the vesicle and calcium channels in these two different synapses, where they are closer to the vesicles and where are they further away.